everybody. We are very happy to continue our conversations and presentations with GERON, a pharmaceutical company which is bringing out a new treatment for our patients. And we have a pleasure today with Faye Feller and with Jeff, who will bring us through a session. Welcome both. Thank you very much for finding time to talk to us and to the patients. Maybe you present yourself, what is your positions and what you're doing.
Sure. Thank you, Peter, and the Global MPN Foundation for having us here today. I am Faye Feller. I am a medical oncologist and hematologist by training. I work at GERON Corporation as the Chief Development Officer. I've been working here approximately 6 years, leading the clinical development team and running all the clinical trials, developing Imetelstat, our telomerase inhibitor, which you may have heard about during the last session from our lead of medical affairs, Christian Diamano. I'll turn it over to Jeff to introduce himself.
Thank you, Faye, and hello, everyone. My name is Jeff Berjunas. I have a PhD by background as a scientist, and my role here at GERON is Executive Director of Medical Capabilities. I've been here a little over 3 years, and one of the many things that I do here at GERON is to oversee the partnerships with our many patient advocacy group partners, such as Peter and the Global MPN Scientific Foundation, and really trying to find like-minded partners who really want to improve the patient experience and ensure that patients really understand the landscape and how science is progressing and how it may help in the treatment of diseases where there really is a need. So with that, I'll hand it back to Faye and Peter.
Yeah, well, I will not talk afterwards. It will be your turn, totally. We are in contact with you since many, many years, and I have been following you, how you made your way through the different phases. I know it's a very long way, and it's also an expensive way, obviously, but you have been step by step, you have been getting ahead, and I think the results which you're obtaining are every time better. So we are very, very much interested to listen, how does your product work? What are you doing? And I think it is very important that we as PAG, that we can give that information to patients. It cannot be as early enough. They have to know, the big work which you're doing, the the attention which you give to all this, and your sacrifices which you're making to bring a product like this up. So for this, I wanted to thank you in the name of all the patients. We depend on you in order that our symptoms are getting improved, and we could maybe finally get a treatment which is chronifying really our illnesses. So thanks for all that work which you're doing. And now please, Faye, I give you the word in order that you can explain us what is your treatment.
Sure. Thank you. I'll begin the presentation, and while we pull up the presentation, I'd like to thank you and the work that you all do with the patient advocacy, because it is important that we keep patients in mind when we do what we do in terms of developing clinical trials and testing new new treatments, because often in the day-to-day we don't see patients, and we do our work, but it's important to know that at the end we're doing what we're doing in order for patients to feel better, to live longer, and to just conquer their disease and manage the disease better. We often need to take a step back and be reminded of that, and partnering with organizations such as yours helps us keep that in mind. So thank you for the work that you do and having us here. With that, I will give you an update about Imetelstat. So firstly, a disclaimer. The use of Imetelstat in myelofibrosis is investigational and not approved by any regulatory authority. Safety, efficacy, and mechanism of action of Imetelstat in myelofibrosis is not established. So when we're speaking about myeloproliferative neoplasms in relation to drug development and our compound imetelstat, we're focusing a bit on myelofibrosis, but we're keeping in mind that myelofibrosis can also develop after essential thrombocytosis or polycythemia vera. So the three main diseases within, myeloproliferative neoplasms that aren't um, BCR-ABL associated are all linked together. ET, PV, MF, and can all PV and ET can develop into MF, and all can transform into AML. So these are all progressive, life-threatening diseases, and whether primary MF or secondary MF. On the relatively rare side and considered orphan diseases. And MF is characterized by fibrosis in the bone marrow. The bone marrow is the factory for all the components of the blood. And fibrosis is essentially a laying down of non-cellular material in the bone marrow. So somewhat like cement and other non-functional material being deposited in the bone marrow um, instead of materials that can help the factory continue to function. So it becomes a dysfunctioning factory. And what happens is the spleen begins to take over as a backup factory, but not a very efficient one. And the spleen enlarges. All of these things lead to symptoms and a decreased quality of life. The symptoms are mostly fatigue or being tired. The big spleen leads to belly discomfort, abdominal discomfort. There's also night sweats and bone pain. So MF is a different experience for each patient, experiences it differently, but these are the common symptoms that can occur. A main pathway when we look at the cells that are affected in myelofibrosis, one of the main molecular pathways that's involved is the JAK inhibitor pathway. So JAK inhibitor is the JAK-STAT pathway. Apologies. So JAK inhibitors were developed in an attempt to shut down all the mechanisms that caused the abnormalities in myelofibrosis. And JAK inhibitors are effective. Ruxolitinib has been approved now almost 15 years and is a standard of care. It helps reduce the spleen. It helps patients feel better. However, many patients do discontinue due to toxicities, or it stops working as effectively. And there are a need for additional therapies. Other JAK inhibitors have been developed, such as momelotinib, fedratinib, and pacritinib, and each of these can be used in certain circumstances. Pacritinib is indicated for patients with lower platelets, and momelotinib for those with anemia, and so on and so on. However, there are no therapies that are currently approved that don't focus on this JAK-STAT pathway. And we know because patients continue to have disease while on ruxolitinib or another JAK inhibitor, that JAK pathway inhibition or using these medications is not the only answer to treating this disease. And there's a need for additional therapies. We also know that once patients fail these therapies, survival and quality of life is quite poor. Shifting topics a little bit, we know that in myelofibrosis, another pathway, and actually in most cancers, um, telomerase is highly expressed. So what that means is telomerase is an enzyme within cells that allows the cells to survive. In normal cells, in the everyday functioning healthy body, telomerase is not expressed at high levels. Cells die and at a normal, regular pace. However, in order for malignant cells or cancer cells to maintain immortality, they take advantage of the telomerase enzyme, and telomerase is overexpressed in order to maintain that immortality and to live longer and maintain that cancer phenotype or that hallmark of cancer that it allows it to live longer. And that's true for myelofibrosis and myeloproliferative neoplasms like ET and PV also as well. They have a high level of telomerase that we see in the cancer cells, but not in other cells. This makes telomerase a really good target for therapy. So, sort of like JAK inhibitor therapy, we have telomerase inhibition. And that's what GERON has been focused on. So, we developed a telomerase inhibitor called Imetelstat. And when this inhibitor binds to the telomerase enzyme, it leads to death of the cells within where it is affecting. And that causes death of the malignant cells or the cancer cells specifically, because those are the ones that need the telomerase to live. And that is specifically true for blood cancers like MF. Next slide, please. So, going back a little bit, we know a couple things about patients with myelofibrosis that lead to a poor prognosis. We know that anemia correlates with a decreased survival, and that increased reliance on red blood cell transfusion correlates with a decreased survival. So, unfortunately, discontinuing a JAK inhibitor, having anemia, relying on blood cell transfusions, all of these factors lead to worse and worse outcomes. Next slide, please. And additionally, we also have to keep in mind in this day and age, there's a significant burden of myelofibrosis on the patient, especially if they're transfusion dependent. There's a lot of utilization of healthcare resources, visits to the doctor, time out of your day, inability to participate in the activities and functions of life that you would normally enjoy, in addition to, costs. So putting the pieces together, knowing that there is a high unmet need in the treatment of patients with myelofibrosis, especially once they are no longer responding to JAK inhibitors, or JAK inhibitors are no longer effective treatment, and knowing that these patients have poor outcomes and a high burden of symptoms and morbidity and mortality. Then, on the other hand, knowing that telomerase is overexpressed in the malignant cells and we have a telomerase inhibitor, we underwent a phase 2 trial to see if imetelstat had efficacy in patients with myelofibrosis who had been treated with a JAK inhibitor and relapsed or were refractory to the JAK inhibitor. So this phase 2 trial, the goal of the trial was really to assess which dose of imetelstat was more effective and more tolerated in this relapsed refractory JAK inhibitor myelofibrosis patient population. So we looked at 2 doses, 8.9 milligrams per kilogram. Imetelstat is an intravenous, so it's an infusion, given every 3 weeks. And the other dose is 4.4 milligrams per kilogram, also intravenous, every 3 weeks. We had 59 patients in one arm, 48 in another, and we looked at co-primary endpoint. So at the same time, both symptoms and spleen. These are common endpoints for clinical trials in myelofibrosis that were established by JAK inhibitors, primarily ruxolitinib. So that's the benchmark by which we measure all other therapies, even if they're not JAK inhibitors, to see if they're effective. Next slide, please. There's a bit of detail about the inclusion criteria. I think it's important here that our patients that were included on this trial had really had a lot of treatment with JAK inhibitors, a long time, maybe more than one JAK inhibitor, and they had big spleens, very symptomatic. So a patient population that had a high need for something different, a new way of treating and a new way of looking at the disease. Next slide, please. So here's what we saw basically in one slide. In terms of the symptom response on the higher dose, we saw a relatively good symptom response. 32% of patients felt better at week 24 by the benchmark of 50% improvement in symptoms. And for a patient population who had already had a response to JAK inhibitor, this is a pretty good result, pretty good outcome, 30%. If we lower the bar a little bit and we say, how many, felt better at any time, not just at that week 24 time point, 50% of patients, 52.5%. So even better if we widen the criteria for response. And when we look, was it just the symptoms of, fatigue and night sweats, or is it just the spleen symptoms that improved? No, it was all the symptoms across the board that were on the questionnaire that improved. So a decent symptom response on the 8.9 mg per kg. However, in terms of the spleen, we didn't see much of a spleen reduction, to be honest. Most patients had some amount of spleen reduction, which you can see on the graph, but a significant amount of spleen shrinking, not very much so. And in the end, we saw better responses in the higher dose than in the lower dose, which was really the objective of the study, which, tell us which dose should we use for the next trial, and really where do we think imetelstat can have the best result? Is it with spleen? Is it with symptoms? Or is it with something else? Next slide, please. So it turns out we think with imetelstat, the answer is with something else. It can have the most effect. When we looked at one of the other endpoints of survival, we saw that in this small group of patients, the survival for the patients in the higher dose was about 30 months. And when we looked in the literature and spoke with myelofibrosis physicians and experts, they informed us that patients, once they discontinue ruxolitinib or other JAK inhibitors, the survival is about a year. So for us, it was remarkable and notable that 30 months was quite different than about a year. Granted, this was a small number of patients. We had no control arm, which means we didn't compare against patients who didn't receive imetelstat. So we decided to test our hypothesis, or to test the idea that imetelstat improves survival or improves how long patients can live. by doing another study comparing our dose, our imetelstat treated patients to those who had been treated at a cancer center in the United States. I can't remember if we have a slide about that, but I'll go through this slide and then I'll talk about that. So this is basically a summary of what we saw in that phase 2 study. So you can see the orange box shows that the higher dose was more effective and the safety profile was about the same. I'll talk about that a bit in a bit. And really the most impressive signal is what we say, but really most impressive efficacy finding was a 30-month overall survival. And then we also saw some other interesting things. We saw improvement in fibrosis, so that, cement and junk deposit in the bone marrow, we saw that getting better in about 40% of patients, which is surprising also, because that is something seen mostly with transplant. We saw some reduction in mutations, which is a sign or a possible sign that the abnormal malignant cancer cells, are reducing over time. And we saw improvements in anemia and transfusion dependence. So all these, not very, are pieces of a puzzle that implied to us a possibility that imetelstat could change the natural history of myelofibrosis and improve patient survival overall. So before I go into this, I'm going to just say a quick word about what we did in order to help us feel more confident about running a study with overall survival as primary endpoint. We did what's called a real-world evidence-based study. We took data from a cohort of patients or a group of patients who were treated at the Moffitt Cancer Center in Florida. And we made sure that they were similar patients to who had been enrolled in our phase 2 study. So we sort of pretended that they would have been enrolled as a control arm or an arm that got just regular treatment and not imetelstat. And we randomized them retrospectively, not prospectively, though, looking back, and matched them with the imetelstat patients on the higher dose, the 8.9 milligram per kilogram arm. What we saw when we did this in a statistical way is two important things. We saw that the patients at the Moffitt Center, when they discontinued ruxolitinib, their median survival was about a year. So this confirmed what we had been told by the myelofibrosis providers and what we saw in the literature. And the second thing we saw was that the survival for the patients treated with imetelstat was, again, 30 months. So it verified the results that we had seen in the phase 2, and it verified potential for an improved survival with imetelstat treatment. And that gave us the confidence to run our phase 3 study, which is now ongoing. So this is the IMpactMF phase 3 study. It has a very similar patient population to the phase 2, patients who have been treated with JAK inhibitors and are no longer benefiting from that treatment. There are about 320 patients randomized to the high dose of imetelstat, 8.9 every 3 weeks, or best available therapy, whatever their doctor decides is the best for them out there. That's not a JAK inhibitor, because they've had JAK inhibitors and the JAK inhibitor is not helping them anymore. So it could be something like Hydrea, hydroxyurea, anagrelide, things like that, steroids, or androgens. And we're primary endpoint is overall survival. And this is important because that's where we saw the signal in our phase 2 study. So that's where we want to test our hypothesis. And we are the first study in myelofibrosis to look at overall survival as an endpoint. So unlike studies in acute myeloid leukemia and some solid tumors, there's never been a study in myelofibrosis done before looking at overall survival as a primary endpoint. Next slide. We're really looking forward to the results of the study. So again, it's very similar criteria to the phase 2 study. We have patients who have symptoms, large spleens, and really are no longer benefiting from any treatment with JAK inhibitors. And as I mentioned before, well, in terms of safety, because imetelstat is active in the bone marrow, we do see hematologic toxicity, meaning we see low platelets, low neutrophils in the majority of patients to a high grade. What's interesting is we don't see the clinical consequences of these. So when neutrophils are low, we expect infections. We don't see those. When platelets are low, we expect bleeding. We don't see a large amount of that. So patients need monitoring when their counts are low, but the hospitalizations for low counts or infections or bleeding, those don't occur with imetelstat. We see that the cytopenias or the low counts are short-lived, meaning they occur for about 2 weeks and they're reversible. We also see some infusion-related reactions, but with steroids and antihistamine pre-medication, those are mitigated. Next slide, please. So zooming in a bit more on the symptoms, this is a graph of what I mentioned earlier, in that if we look at the purple bars or the higher dose, this is each symptom on the questionnaire that patients were taking or answering every day. We can see that patients improved. So the higher bar means an improvement in their symptoms or a reduction of their symptoms in each symptom: inactivity, pain, itchiness, abdominal pain. Next slide, please. Shifting gears again a little bit, we have our phase 3 study in relapse refractory, previously treated with JAK inhibitor patients that, we're anxiously awaiting the readout. But one of the principles of drug development in oncology is, if you think you have a drug that works well and can help benefit patients, especially if you think it can increase lifespan or change the course of the disease, the earlier you start treatment in the disease course with that drug, the better it is. So we quickly started a study to see, can we combine imetelstat with ruxolitinib safely, so that we can move it up or start treatment with imetelstat before patients start failing, or before the drug starts failing the patients. And could that be done first? We have to figure out if that can be done safely, and then we have to figure out if that has any efficacy. So to answer the first question, can that be done safely? We did a phase 1 study, IMproveMF, or we are doing, it's still ongoing. We completed the dose escalation part. So we started with a very low dose of imetelstat and used 4 different doses. Slowly, just a couple patients at a time, we tested small doses, then higher doses, to make sure, in combination with ruxolitinib, whatever dose the patients were on, to make sure that taking ruxolitinib at the same time as taking imetelstat was safe and patients didn't have any severe side effects. We also know that ruxolitinib can cause counts to go down and imetelstat can cause counts to go down. So we really were on the lookout to make sure that platelets, neutrophils, and hemoglobin didn't drop too low when taking the combination. And what we found was even at the highest dose, the 8.9 milligram per kilogram dose, the combination of imetelstat and ruxolitinib was pretty well tolerated. Patients did not have bleeding, they did not have infections, and it seemed that this is a safe combination. So we're moving forward with the highest dose and adding more patients so that we could see if there's any efficacy, if spleens get better, if symptoms get better, if we see any changes in mutations or other signals like that. So this is an ongoing study. Next slide. So unlike our other studies, this has patients earlier in their treatment course, right, is the main point of this slide. But they also have active symptoms or spleen. Here you can see a summary of what I said is that basically their side effects were mostly anemia, so a low hemoglobin, and neutropenia, low neutrophils, low white cells, but you can see low rates of that, 24%, 18%. Next slide, please. Excuse me. We do have some early data looking at symptoms. And so here, unlike the other graph I showed, the bars going down means symptoms are getting better. And you can see the purple is the highest dose again. So the lower the bar, the better the symptoms. But even on low doses, patients' symptoms were improving. Again, this is a small number of patients, but it looks like something's working. Next slide. And this is the data for spleen. Again, very early, low number of patients. But the blue is the second highest dose. This is early data. We didn't have enough patients treated at the highest dose yet to show the data, but we'll be updating this in the future. So overall, we believe that imetelstat as a inhibitor of telomerase, its unique mechanism of action because it specifically targets the part of the cell that drives the malignancy and immortality of the cancer cell. It's different than a JAK inhibitor, and that can potentially provide benefit in terms of disease modification for myelofibrosis patients. And we'll learn much more when the phase 3 survival study reads out and add to that knowledge with our phase 1 combination study and whatever follows after that. So we're excited and hopeful. Thank you.
Well, thank you, thank you very much. Very impressive. It's really a very interesting information you gave us here. So basically, your first trial is just the product itself, and now the second phase, you're trying to combine it with Fruxo. You were talking about also the symptoms. I didn't see very much about fatigue. Is that something which you looked at it? Is it improving? I saw some symptoms, but for example, fatigue, I didn't see very much. Did you work on that also? Did you try to find out?
Yeah, so interestingly, the main questionnaire that we used on the phase 2 study, the standard analysis for that, even though fatigue was collected as a question, the standard analysis did not remove fatigue from that calculation of TSS. So we have the fatigue data. I just didn't show it. But in general, we have shown an improvement in fatigue. And what I can share is that our drug has been approved for the treatment of lower risk MDS and for the treatment of transfusion dependency. So it helps to release patients from being reliant on transfusions, lower risk MDS patients. And we, in our phase 3 trial on that, with that patient population, we did see an improvement in fatigue that
Okay.
was a bit unexpected, despite those patients receiving less transfusions. As we know, transfusions help fatigue. The last thing I will say is our phase 3 study. We are using fatigue within the total symptom score, so
Yeah,
we'll have
because
those results
it's
as part of TSS.
yeah, the symptoms itself. This is more and more of a major worry from a patient. We have products. I'm very happy that we're getting out from the only cytoreduction products. You're really going a fantastic way. It looks to me like if this way could be followed, we are close from not curing, but at least from um, trying to stop it, to have a certain stop in myelofibrosis also, but it will not follow a chronification of myelofibrosis. Do you think that this will come? I mean, you talk about the survival. Uh, you know, I mean, you're starting with patients, I think, with a very, very low count of platelets. So something, I think, below 50,000. So, the survival obviously is not the best one. But if this medicine could be taken earlier, could not that be the beginning of a way to bring us to have our illness to be stopped, not cured, but at least stopped one or the other way.
Yeah, I completely agree, Peter, and I think that it's an unfortunate quirk or history of drug development that we have to start with single agent therapy in a relapse refractory patient population and demonstrate benefit there before we can move to earlier lines of treatment. But I think the ultimate goal is, if we are a disease modifying therapy, is to move it up front so that patients can derive the most benefit as early as possible.
Yeah, I mean the the way how you're attacking the problem. Um, I'm a patient. I'm sorry. I don't have really any background from chemistry or the work which you're doing. But for me, I take things very, for me, they look very easy when I see that. But shouldn't it be that, this way of attacking the problem, that you inhibit the cells, the cancer cells to multiplicate and they start to die? Uh, that should be able to be done already with PV and with ET.
hmm We did have early studies with ET. There's actually an article published in the New England Journal of Medicine about, I want to say, 10 years ago, that showed that in ET patients there was 100% response rate with Imetelstat.
Mhm.
We put that development on hold at that time. We're a small biotech, and decided to focus on myelofibrosis due to the unmet need at that time. Now we're growing, and we'll see what the future holds. But there is potential for ET and likewise PV.
And are you starting trials, or I didn't understand, but you're planning to do that?
We'll see where it goes. We're still, small, but we're growing. So we'll
I
see. I
know.
think we have to wait to see how the MF study reads out, the phase
Yeah.
3. I think if that reads out positively, that will, really drive how we approach the MPNs in general.
Do you know whether your treatment is affecting the hemoglobin value? Because we know Jakavi, many patients has a side effect that the hemoglobin value is getting down. So it's somehow affecting the anemia. Uh, do you know something about this? Is it affecting hemoglobin account?
Yeah, it's interesting because we do see anemia reported as an adverse event, but we also see about a 25% transfusion independence. So I think initially there may be some anemia as a toxicity, but over time, as the marrow improves and recovers, hemoglobins can rise, leading to transfusion independence.
Yeah.
So if we think about theoretically the drug acting on the bone marrow, first, reduction of malignant cells may cause neutrophils, platelets, even hemoglobin to go down. But then once the marrow recovers, things can
It
go
will
back
go
up.
up again. Yeah, 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 yeah. I mean, it's fantastic, when I only think about it, that we would be able to reverse, the damage in the bone marrow, and that,
Yeah.
the fibers will be gone, and a normal function of a more bone marrow would be possible. This is
Yeah.
it's like a wet dream, but I, I think you make it possible. I'm very, very
Yeah.
happy.
I
And
would also refer you to the New England Journal paper about myelofibrosis. I failed to mention, my apologies, but like there was a small pilot study done at the Mayo Clinic, also about 10, 11 years ago, that showed complete reversal of fibrosis. And those pictures, pathology pictures, are in the New England Journal of Medicine. And it is kind of dreamy.
yeah, it's the goal. It's at least, keep it in
Yeah.
front of you, and this is where I have to this is what I have to reach.
Yeah.
But
It was inspirational for me as a developer, drug
yeah,
developer.
it's really, it's I just can tell you that we have I've just now I have a sort of a steering committee of MF patients from all over the world, and we're trying to bring together the unmet needs for a MF patient. And when you listen to the stories of each of these patients, what they have to suffer due to the side effects, it is simply impressive. It's depressive also. Because most of the people were losing their jobs. You have problems that your head is dizzy. You cannot think in the right way anymore. Concentration problems. Besides of what we know about the fatigue. I know patients, they simply can't get out during 4 or 5 days. They're in bed, incapable to do something. So it affects very much, the population. We are not many, but
Mhm.
they are all suffering tremendously from a side effect. So I'm very happy to see that more and more the side effect is coming into the game, and that you're asking and checking the side effects during your clinical trials, which I think is tremendously important, because I think very soon we might do together a session where we have patients with you, where you can listen to what patients are explaining. It is a very hard life for the patients to live with all the symptoms, and we are very thankful that you're going this way, and that you keep on battling despite of all the problems. And I know it's a very long marathon which you have to go there and to do it. Um,
Yeah.
I do have there are some questions in question and answer but all the questions are coming from an anonymous participant. It's my rule, but I don't give questions which are coming from an anonymous person because I don't know the origin. We are, despite of the fact that we are using a lot of intelligence, we also know the danger which is coming from the artificial intelligence, and they manage to get involved in this kind of these meetings, and they're just putting questions for their own interest. And I have seen the questions, and I will not ask them the things which they're doing. So, but there are 5 or 6 questions, but they're all from anonymous. And I'm sorry, but it's our rule that we don't reply on that because you can give your name, and I think this is how it should be done. Well, from my side, I only can thank you very much. We will collect the questions which are coming up. I know for sure we normally get through our websites and social media, we're getting questions about what you have been telling us, and we obviously will come back to you and we will tell you these questions. But it was extremely informal, very, very clear. You brought up a very patient-friendly language, which is, I know, very difficult to do that in your complicated world. But just thank you very much, and don't forget you do it for us, people, patients, like me, which we have to sometimes go a difficult way. Yeah. So
Yeah.
thank you. That's great. And, we keep on talking, what we do next between us. Yes.
Thank you. Yes, certainly looking forward to more collaboration. And I think in general, the more opportunities industry has to collaborate with patient advocacy, the better. Industry, academics, patient advocacy, regulatory, we should all be meeting together as often
Thank
as we
you.
can
Well,
to
for
improve.
any need you have to get the patient information, we are here for you. We can provide you, and we would love to give you as much as information as you need, because this is for your work which you're doing. I think this is vital.
Yeah, appreciate
Also,
it.
also thank you
Thanks
to
for having
Jeff.
us.
Yeah, thank you, Jeff, that you were here today. It's a pleasure. And
Always a
we will be in touch, and we'll talk our next steps. Yeah. Okay.
absolutely.
Thank you very
Take
much.
care.
Yeah. Okay. Have a nice day.
Have a good
Thank you very
evening.
much. Thank you. Bye-bye.
Thank you all.
Bye.